Okay, so we finished the paper. One of the main reasons why we went to see this one is because we had extra lectures on Saturday. Not everybody could be there, so we took a little bit different way from the normal course. What we are going to continue is to read the paper that we read before this RNA methylation paper, the one about the biological clock in bacteria. And if you remember, there was something that needed to be explained briefly on the page 235.
Which paper?
Yeah, this one. It's
Picture of the mammalian circadian.
no. The one timing the day. What
Oh.
makes bacterial clock tick? And if you remember, there was this sentence: the transcription translation feedback loop controls transcription and translation, whereas the clock specific nanocomplex. Do you all? I have no copies.
I shall put it online.
I can probably go and make a copy quickly in the.
I can get it online.
Can you?
Yeah.
Do you have a? Really?
Sci-Hub, where you can access everything for free.
Really?
Yeah.
Can you access this paper for free
Yes.
with your student account?
No, it's called Sci-Hub, and this is like the name of any paper, and you get it for free.
You know that as well? Do you know Cyclope as well? Yeah, you have students, so you can. Cyclope?
Sci-Hub.
I don't know Sci-Hub.
I don't. Do you know Cyclope? What the heck? Can you show us?
Have you not seen this?
What the heck?
It's absolute. It's saved my ass so many times when it loads.
Cyclope.
Yeah, and it's a Russian site because she got really bored of having to pay for everything. So like everyone puts their access keys on, and now
So you get nature and science and
everything free. Yeah, you just need to enter the title and.
Russians.
Russian.
Russians.
With Kyoto University student ID, I can access.
Yes.
Yeah, but I can do it on Glasgow as well, but this is easier.
All right, I haven't heard.
What's the title?
I haven't heard. Timing.
Illegal, but
I want to see. I don't believe it. Timing the day. What makes bacterial clocks tick?
oh, I spelled it wrong. So it's not gonna work. There you go.
Oh yeah, this is. Oh my god. And it's very fast as well.
Yeah.
They do all sorts of things now. The Russians. They even like influence Americans' elections. All right. Okay. So. Right, I forget about this. So this is page 235. It's the the left column that starts cycles within cycles, robustness and synchrony. And the last sentence of that left column was, the transcriptional translation feedback loop controls transcription and translation, whereas the clock-specific nanocomplex, which is composed of Ki-A-B-C, is a mass action biochemical oscillator. And mass action, we had a little bit of a hesitation there, if you remember. So have any of you looked what was mass action oscillator? Actually, mass action, it's a very common chemical name. It's one of the most simple type of reaction where the rate of the reaction is dependent only upon how many products and reactants that you have. So it's a very simple chemical reaction. So that's what they are saying is Chi A, Chi B, Chi C. It's a mass action biochemical oscillator, so it's a biochemical oscillator that depends on the abundance of its members. Okay, one of the very simplistic chemical reactions. That's simply what it means. Okay, like for example, if you have this kind of thing, x plus y forms a complex xy, then you know that the formation of a complex will depend on the abundance of each xy in the complex. Okay. So very standard chemistry. Okay. But where did we read? What is the end of the reading?
On page 236, we finished the first paragraph. The sun also rises by the bacteria.
So, or is it possible that a circadian clock could be composed of Chi B, Chi C, or even just Chi C alone? Okay. So I will read the next paragraph. In this context, the example of purple bacteria may be instructive. Purple bacteria are globally distributed members of the proteobacteria that frequently inhabit soil and water environments together with cyanobacteria. Tests on circadian rhythmicity have been reported for two species of purple bacteria, Rhodobacter spheroides and Rhodopseudomonas palustris, which both have homologs of Chi B and Chi C. R. palustris does not sustain rhythmicity, as monitored by nitrogen fixation and the phosphorylation of Chi C in constant condition, which is a defining characteristic of circadian rhythms. In R. spheroides, rhythms of gene expression were detected 21 hours under aerobic condition and 11 hours under anaerobic conditions. But other defining circadian characteristics, such as temperature compensation and entrainment, were not reported. Okay? So very strangely, not all bacteria have a molecular clock, as far as we can analyze. Okay? So they are not all like cyanobacteria. They have Chi B and Chi C, but something is missing. Okay, maybe Chi A. Who wants? Is it okay? This paragraph is fine. Don't hesitate to ask questions. Nitrogen fixation. You understand nitrogen fixation? What is nitrogen fixation? Minna wakaru? Nitrogen fixation. What about you two? The ladies understand. What about you two? Wakaru? Nitrogen fixation. Do you understand?
I have heart.
So it's actually using inorganic nitrogen and fix it into organic molecules. Okay. Like carbon dioxide fixation is using carbon dioxide to make sugar, for example, in plants. Questions for this paragraph? So the last sentence, in R. spheroides, rhythms of gene expression were detected 21 hours under aerobic condition and 11 hours under anaerobic conditions, but other defining circadian characteristics, such as temperature compensation and entrainment, were not reported. That just means they were not found. It might have been there, but they were not found. Okay. And actually, it's the same for many bacteria. They may have rhythms, but so far, some have not been detected.
How is it different under aerobic compared to anaerobic?
The metabolic pathways that they are using is completely different. Under aerobic condition, you can use oxygen as the final oxidant. But if you don't have oxygen, then you have to rely on other things to actually produce your energy. You can do glycolysis and all these things. And so this will actually cause dramatic differences in your metabolism. And so this is why sometimes this will also affect your rhythms. Okay? This is only rhythms. It's not a clock or anything. Rhythms were detected, but whether it's a clock or not, it's not. Okay, so we can go, if there are no questions, we can go. We read the next paragraph on your Sci-Hub.
Okay. Recently, the daily profiles of Chi-C phosphorylation during light-dark cycles, continuous darkness, and continuous light were compared among S. elongatus, Orococcus virilis, and R. palustris. C. elongatus, which contains Chi-A, Chi-B, and Chi-C, exhibits daily rhythms during light-dark cycles that are sustained in both continuous darkness and continuous light. By contrast, P. marinus and R. palustris, which contain Chi-B and Chi-C, exhibit daily rhythms of Chi-C phosphorylation during light-dark cycles that dampen rapidly under constant conditions. We previously proposed that the timekeeping system in R. palustris is a proto-circadian oscillator that does not persist under constant conditions. This hypothesis is consistent with the observation that P. marinus, which lost Chi-A, also does not sustain robust rhythms of gene expression or Chi-C phosphorylation under constant conditions. Taken together, the data support the hypothesis that all three Chi proteins are necessary for sustained circadian oscillations in bacteria, and that Chi-BC-based and Chi-C-based systems might be capable of hourglass timekeeping, but not a persistent circadian oscillation. Given that the sequestration of Chi-A promotes synchrony in C. elongatus, Chi-A was perhaps an evolutionary innovation of cyanobacteria to sustain oscillations, which resulted in their robust persistent rhythms.
Okay, thank you very much. So that's the synthesis of what we have read so far. And one of the main motivations of me selecting this paper is that why do we have clocks? Okay, this is the next point in this paper, is that rhythms were found in bacteria, rhythms were found in eukaryotes, human, plant, reptiles, you name it. So rhythm seems to be something that is universal. It's not in everything, but it seems it can be found in every single, at least in a few members of every single phyla. Okay? And of course, it's very difficult to study that in human and to study that in mouse, but with bacteria, it's very elegant to actually study whether having a biological clock may actually mean that you're actually more competitive than other organisms. And that's the next point of this paper, which I think is quite kind of interesting. Any question on the previous paragraphs that you just read? That should be okay, I think. No? If there are no questions, then Nguyen-san, you can read the next.
Timekeeper evolution and new criteria for testing adaptive fitness. Speculations about potential adaptive values of timekeeping in bacteria are thought-provoking, but have there been any rigorous tests of the fitness advantage? In cyanobacteria, strains that have a functioning biological clock outcompeted arrhythmic strains in rhythmic environments, for example, like life cycles, whereas in constant environments, for example, continuous life, rhythmic and arrhythmic strains comparable rates. Moreover, among rhythmic strains, those that had endogenous free-running periods that closely matched the periods of the environmental cycle had the greatest fitness advantage. Competition experiments are sensitive tests of fitness, and they clearly indicated that the cyanobacterial circadian system interacts with the environment to mediate an adaptive advantage, especially as flock mutants could outcompete wild-type cells under appropriate conditions.
Okay, thank you very much. So that should be understood, I think. That's pretty easy. No difficult terminology. Okay. Okay, then let's continue. Next, please.
More recently, the fitness benefits of a timekeeping program have been assessed in our palustries. As mentioned above, our palustries does not exhibit sustained oscillations under constant conditions. Nonetheless, does Chi-C carry out an adaptive timekeeping function in this purple bacterium? To address this question, the growth rates of wild-type R. palustris and Chi-C knockout strain were compared in single-strain cultures. Both strains grew at the same rate under non-selective continuous light conditions, but in the 24-hour cycle, 12 hours of light and 12 hours of darkness, the Chi-C knockout cells grew slower than wild-type cells. Therefore, Chi-C enhanced growth in the 24 hours light-dark cycle, but not under continuous conditions. Importantly, additional experiments with a 1 hour light and 1 hour dark cycle ruled out the reduced amount of light exposure during cycles of 12 hours of light and 12 hours of darkness as a confounding factor. Therefore, the Chi-C gene of our palustris has an important effect on growth and fitness in 24 hours cyclic environments, but not in non-24 hours environments. Continuous conditions with cycles of 1 hour of light and 1 hour of darkness. This result strongly implies that the fitness advantage of Chi-C in our palustris is related to adaptation to environments that have a 24 hours periodicity. Growth experiments similar to the ones described above can be considered as a new criteria to assess whether Chi-C is involved in an adaptive 24 hours timekeeping mechanism in any bacterium.
Okay, thank you very much. Questions for this one? So, it's relatively easy to understand that, of course, the presence of a clock might be advantageous in a rhythmic environment. If the environment is not rhythmic, the advantage of a clock might not be very strong. So this is why usually in a rhythmic environment, the strains with a clock are usually better than strains without. But if the environment is continuous, then actually there is maybe no very big benefit. By the way, there is an interesting thing that comes into my mind. So, when you study the circadian clock in mouse, when you have mice or rats, these species are nocturnal, so they live during the night. And when you want to raise them in a constant environment, which usually should you choose? What kind of constant environment? Of course, temperature should be constant, food should be provided all the time, but what is the main factor in terms of light? What do you choose usually? Do you remember? Yes, constant darkness. But what if you are studying monkeys or plants? First of all, monkeys. What kind of environment should you raise your monkeys in? Diurnal monkeys, like marmoset or whatever.
Constant light.
Constant light, yeah, constant light. Usually, diurnal animals in constant condition is constant light, and nocturnal animal, it's constant darkness. Of course, experiments have been done with mice using constant light, and that's very interesting because light for a nocturnal animal is actually quite powerful. And some strains of mice actually become completely arrhythmic in constant light. Because if you want, light is always there, always stimulating the clock, and some strains of mice are extremely sensitive to that. And some mice, the rhythms completely destroys, and you have strange things happening like 12 hours rhythms and some sort of stochastic rhythms. So some species of nocturnal animal cannot really cope with constant light. So there are a few interesting discoveries with that, but not so many experiments are done now because it's not very well. It could be considered to, always having light at night and things like this in humans, but.
Is it the other way around? Like if we were in constant darkness, would our rhythms go haywire?
Like, because you said the mice in constant light go mad.
Well, the rhythms do. Like if we were in an experiment, we were playing constant dark, would our rhythms also go?
I don't think it has been done. There were weird experiments done with humans in which, volunteers were left in some sort of mine or caves and things like this. But to my knowledge, they have not shown these kind of things, that the rhythm, all the studies that I am aware is that when humans are left in constant condition, they just continue with their free running period. And I don't think that they continue the study for long enough to actually see these things happening. So, you can imagine it costs a lot, right? Right, you are going to be in this cave for 3 months. This is is that all right with you? So studies with humans is a little bit difficult. So this is why actually our professor here, he is retiring next year. The last thing that he wanted to do, and he is currently doing, I think is great, he is studying monkeys. And because they are closer to humans. So it's not really feasible to do that with humans, but with monkeys you can sort of. So he's doing jet lag experiments and things like this, but yeah. So, okay. Then we can go to the next paragraph.
What might be selective pressures that led to the evolution of timers versus oscillators? In a sufficiently regular environment, a damp oscillator or even an hourglass timer might enable adequate anticipation of daily changes in the environment. Some chronobiologists have questioned the necessity of a sustained circadian rhythm, as circadian clocks evolve in a cyclic world, not continuous conditions. It is the mechanism of entrainment that involved and not self-sustained oscillations. Nevertheless, among eukaryotes, daily biological clocks are almost universally self-sustained circadian systems. How and why is this the case? Perhaps studying the evolution of daily timekeepers in bacteria may help to address these questions, especially in bacteria that have chi homologs that can be identified and targeted for fitness tests, as shown in Figure 5. For example, a bacterial species that is always exposed to a regular light-dark cycle with a consistent photoperiod and consistent temperature might effectively adapt to a cyclic world with an hourglass timer. However, an oscillator enables several outputs and or flexible phasing, even if the oscillator is damped and requires a relatively consistent light-dark cycle, such as the KI-DC-based system of R. palustris. Finally, in bacteria that inhabit temporally irregular environments, for example, environments with unreliable exposure to light and darkness, inconsistent light intensities, annually varying photoperiods, or irregularly available energy sources, a fully developed circadian oscillator provides the maximum temporal buffering to accurately track the solar day. As an example, seasonality may create very different selective pressures for free-living bacteria compared with gut bacteria. For a free-living phototroph, the differences in average temperature and photoperiod between winter and summer affect growth rates. Gut bacteria may enjoy a more consistent annual environment, but the diet of the host can differ substantially between winter and summer, which leads to drastic annual nutrition cycles that are known to affect the composition of the microbiome. Currently, the model showing figure 6 is speculative. However, the widespread distribution of Chi homologs in various bacteria, which inhabit vastly different environmental niches, prevents a unique set of test cases with which to analyze the evolution of daily biological time.
Okay, thank you very much. And here we have a new term, which I think I've never explained. Photoperiod. What is photoperiod?
Just a light-dark cycle.
It is not. It is linked to the light-dark cycle, but it is not actually.
Like the light. Time of light.
The time of light?
So the time of like we have 8 hours of sunlight.
Yeah. So the photoperiod is the relative duration of night versus day. So in winter you might have 8-16 LD photoperiod. And in summer, you may have a 16-8 LD photoperiod. So this is what it means. And the clock has to adapt to these things. Versus, 16 hours versus 8 hours of light is significant. So maybe the clock evolved to actually be able to cope with these things. So that it's able to track 24 hours regardless of the photoperiod. Light is still important to untrain the clock, but because it's endogenously encoded, then it can track 24 hours, whatever the duration of light. Any questions on this long paragraph? So you can take a look at the figure 6 that they said. It's only a speculation, it's a model, it's nothing really certain, but it's a figure that shows selective pressure and the response of the animal or the living things that has evolved. Well, it's just what they explain. It's just what they explain. So what do you think, if you go to the North Pole, you have days and nights of like months, right? What would you expect these animals to have? Would you expect these animals to have a strong circadian clock? If you think about this paper, what they are kind of saying, that the figure 6 says that in different environments, you kind of theoretically develop different types of timing mechanisms. So if you think if you think about the North Pole, what would you expect animals there to have? And there are people studying, Arctic biology is kind of interesting. If you were to measure circadian rhythms in animals living in these very high latitudes, what would you expect these rhythms to be? Of course, you can imagine their seasonal clock will be very, very powerful. they have very strong seasonal clock. Like even before the beginning of the summer, they will already, all the hormones will change to be able to reproduce and do whatever they do. But what about the circadian clock? Would you expect Arctic animals to have a strong circadian clock?
But they adapt. When they're taken out of the Arctic in zoos, they adapt. So, not in the wild, but they do have one.
What do you mean they adapt?
We've got polar bears in the zoo here, and they have the night 24 hours.
Yeah, but do you think they suddenly... Okay, so one part was correct. Yes, that's right. In the wild, animals there, they don't really have a strong circadian clock. And if you put them in constant darkness, actually they don't have any beautiful rhythms, actually. Because there is no reason for them to have a very strong clock, since the environment they live in is not circadian, it's not daily, okay? But, that's right, if you put them in a zoo, they will show rhythmic activities. But does it mean they adapt by suddenly having a circadian clock?
And it's entrained,
Yes.
behavior adapts.
That's right. They will have rhythms, they will have daily rhythms, but they will not have circadian rhythms. That's right. So Arctic species usually don't have a strong clock, because they're in an environment where it's not really needed, because there is no daily rhythms, okay, most of the time. Any questions on this paragraph? I'm sort of digressing. If not, then so okay, that's also interesting. Daily timing and the microbiome. The relationship between the host and its microbiome has become a central topic in biology and medicine. The temporal dimension of this relationship warrants serious investigation. At the very least, because most animal hosts eat on a daily schedule, the environment that is experienced by the gut microbiota undergoes a 24-hour transformation in nutrient availability. A bacterium that can anticipate the arrival of fresh nutrients might readily metabolize these resources and thus gain an advantage over time-ignorant competitors. Our knowledge that at least some free-living bacteria have daily timekeepers opens the possibility that bacteria of the microbiota have endogenous timekeepers that interact with the biological clock of the host. This blossoming area of inquiry has already identified a gut bacterium, Enterobacter aerogenes, that seems to express circadian rhythms when isolated outside of the host. Interestingly, the robustness of its rhythm is enhanced if melatonin is included in the culture medium. Melatonin is a circulating hormone that is rhythmically controlled by the circadian clock in vertebrates. And these results imply that melatonin could stimulate rhythmicity in members of the microbiota in vivo. In addition to melatonin, other candidates for daily time cues include diurnal cycles of nutrients and circadian body temperature rhythms. Core body temperature oscillates diurnally in most endothermic animals. Microbiota usually means your gut bacteria, okay? Or the microorganisms that live in your gut. Melatonin is a hormone synthesized by which gland in the body? What synthesized? Which? Pineal or pituitary?
Gotta go pineal.
Pineal gland. The pineal gland. So there is a direct link from the SCN, the suprachiasmatic nucleus, to the pineal gland. And the synthesis of melatonin, while it's slightly different in different animals, but the synthesis of melatonin rises during the night and stays high during the night and very quickly goes down at the end of the night. So there is a circadian rhythm in melatonin. Even in constant condition, we make melatonins when it's our biological night, but also it's directly inhibited by light. So during the night, if you shine light in your eyes, melatonin synthesis will quickly go down, and melatonin is very quickly degraded in the liver. So as soon as you get light, melatonin will go down. Okay? And melatonin is one of the main medicine, not in Japan, but it's the main medicine against jet lag at the moment, to try to promote a good night's sleep. Okay? And strangely, mice that are commonly used in laboratories, like it's a mouse called Black 6, C57BL6 mouse. It's a very common strain of mice. These mice cannot synthesize melatonin. Mice in the wild, of course, can, but some laboratory animals cannot. So melatonin research can be a little bit difficult with mice. So you have to choose a strain that can synthesize melatonin. Melatonin is very powerful in humans, but in mouse, again, it's not that powerful. Okay. Even mice that can actually synthesize melatonin, some of them, it's not really clear exactly if melatonin has a strong effect on synchronizing the clock. So, but anyway, in human, it's working. Endothermic animals, you have a definition in the left. It's warm-blooded animals. Do you know what's the opposite of endothermic animals? Ectotherms. Okay, any questions on this one? No, should be alright. Okay, then can continue.
Moreover, if members of the gut microbiota have timekeeping capability, might they release signals that influence the circadian system of the host? The microbiota secretes a vast range of compounds, many of which are bioactive. For example, some bacteria that inhabit or infect mammals can release cytokine-like factors, which, if released rhythmically, could influence the circadian clock of the host. For example, microbial metabolites, in particular short-chain fatty acids such as butyrate, can modulate circadian gene expression in hepatic organoids. Conversely, perturbations of the biological clock of the host affect the composition of the gut microbiome. Several studies have reported diurnal changes in the relative abundance of bacterial phyla in the gut microbiome. Disruptions of the biological clock of the host, either by jet lag or by mutations in the clock genes of the host, alters the proportion of these phyla that exhibit daily changes. Moreover, alterations to diet, such as time-restricted feeding and transition between a regular and a high-fat diet, are known to affect circadian clocks in mice, and they also modify the composition of the gut microbiome.
Okay, thank you very much. So this is very new. These microbiome circadian research, they really started like 2 years ago. This is very, 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 very new area of biology. And it seems, as they say, there is a very tight link between the two. No question? This part is easier than the first part. Okay, then let's continue.
At this time, it seems clear that the composition of the mammalian gut microbiome is temporarily changing, and that changes to the circadian clock of the host affect the viability and relative growth of some members of the microbiome. Wherever there is a feedback from the microbiome to the circadian clock of the host is less well characterized, but it is a fascinating possibility to investigate. Along these lines, Hayes and co-workers reported a remarkable interplay between host and microbiota. And Antibiotic treatment of the host affects rhythmic normalization and adherence of bacteria to the intestinal epithelium, and this in turn reprograms quality and transcription oscillation in the host. Remarkably, these effects occur not only in host intestinal cells, but also extend to the liver, which modulates hepatic secretin gene expression and detoxification. Finally, at least some members of the gut microbiota may have endogenous timekeepers that promote their adaptation to the intestinal environment and to the external environment after they are excreted.
Okay, thank you very much. Should be okay, ne? Then we can continue.
Beyond the gut environment, there are other fascinating examples of host-bacteria relationships that have temporal dimensions, such as the symbiosis between the squid Euprymna scolos and the luminous bacterium Alivibrio fiskeri, formerly known as Vibrio fiskeri. E. scolos has a specialized light organ that is colonized by the bacteria. The luminescence that is produced by A. fiskeri is used by the squid as a camouflage strategy, in which the animal emits light from its ventral surface to mimic the nocturnal moonlight and starlight that shine from above. The luminescence varies during the day-night cycle, with the highest levels of luminescence in the early evening when the animal begins to forage. An examination of the transcriptome of the resident F.A. fiskeri revealed day-night oscillations of metabolism between anaerobic respiration and fermentation. This adaptive temporal interaction between host and symbiont illustrates diurnal reciprocity between host and microbial partners.
Okay, thank you. Those are very weird examples of how tight link can occur between hosts and bacteria. So it might not be so surprising that there is such a link between our microbiome and our clock. To my knowledge, there is no discoveries of any microbiota thing that can influence the rhythm in your SCN. It's kind of okay to understand that there is a link maybe between the gastrointestinal tract and the gut microbiome. But between your central clock and the microbiome, so far there is nothing. Okay? So far there is nothing. And okay, now we can continue with the conclusions.
Conclusions and future perspectives. Although cyanobacteria are an archetypal model system for endogenous self-sustained timekeeping mechanisms in fitness, we have barely glimpsed the monopoly of likely timing mechanisms that have evolved among bacteria in response to various environments and selective pressures. Homologs of the Kai genes are widely distributed among eubacteria and Archaea. Although the cyanobacterial cycles within cycles design is optimized for resilience to internal and external noise, other bacteria might use Kai proteins in different configurations to adjust the daily patterns of their particular environment. There is every reason to believe that new types of timekeeper other than circadian clocks exist and are ready to be discovered. As we expand this research to other bacterial species, new tools will be required to monitor daily rhythms. Luminescence reporters have been crucial for optimally monitoring rhythmic gene expression in aerobic eukaryotes and cyanobacteria. But luciferase reporters require oxygen. Therefore, to monitor rhythms from bacteria that operate in various metabolic modes, the development of automated technologies for microaerobic and anaerobic environments will facilitate the assessment of the generality of timekeeping mechanisms among eubacteria and Archaea.
Thank you very much. should also be okay. So I will continue directly. Understanding the host microbiota relationships have a temporal dimension is an exciting new research area. It is clear that the feeding rhythms and diet of the hosts affect which bacterial phyla thrive in the gut. Signals that emanate from the microbiota reciprocally modulating the circadian clock of the host is a tantalizing possibility, and if found to be true, the implications for chronobiology are profound. As many members of the microbiota are not restricted to the gut, but can also exist in a larger ecosystem that is also rhythmic, a daily timekeeper is potentially useful in both environments. Bacteria that alternate between internal and external environments might adjust the timekeeping mechanism if they relocate, for example, after excretion. For a timekeeper in the regular 24-hour external environment, the precision of cadence may be essential. Whereas in the more variable environment of a host gut, flexibility of tempo may be more valuable. So this is kind of an interesting sentence here that says that maybe some animals may have a clock in a different environment, able to switch it off when it's actually not much needed. So that's kind of interesting. And we are going to finish the last paragraph.
From the broader perspective of chronobiology as an example of evolutionary adaptation, testing simple hypotheses about fitness can be challenging in higher organisms, in which many processes become interdependent over evolutionary time, so the adaptive value of a specific trait, independent of the background, is difficult to ascertain. Bacteria may provide clearer insights into the relative roles of various environmental factors in the natural selection of biological timekeepers. This is not to say that bacteria are simple, that their small and generally streamlined genomes may enable more precise manipulation and measurement of the fitness consequences of variation in traits that underlie daily timekeepers. These factors lead to greater confidence about the functional basis of interactions between trait variation and fitness, because of greater experimental control over both the genomic background and the environmental conditions. Therefore, the adaptive role of daily timekeepers and the selective pressures that led to the evolution of temporal precision, temperature compensation and entrainment may be best studied in bacteria. It is time and timely for microbiology and chronobiology to contribute more to each other.
Okay, so this is obviously written by a microbiologist. So, there is one thing I want to go back, and I want you to criticize one point. It's when they describe the selective advantage of having a clock versus not having a clock in a rhythmic versus a constant environment. So I will read again one of the sentences that I want to find. Yes. To address, so this is on page 237, the second paragraph on the right. More recently, the fitness benefits of a timekeeping program have been assessed in R. palustris. As mentioned above, R. palustris does not exhibit sustained oscillations under constant conditions. Nonetheless, does GAI-C carry out an adaptive timekeeping function in this purple bacterium? This is the important part. To address this question, the growth rates of wild-type R. palustris and GAI-C knockout strain were compared in single-strain cultures. Both strains grew at the same rate under non-selective conditions, but in the 24-hour cycle, the GAI-C knockout cells grew slower than wild-type cells. What do you think? If you want to study the selective advantage of GAI-C, is that what you would do? Of course you can do, but what is the problem? Is there a problem at all? Do you think it's fine?
GAI-C maybe have more roles than just in the...
Yeah, That's true. You are one step faster than I am. So yes, Chi-C knockout cells. Yeah, of course they don't have the Chi-C oscillator, but Chi-C may have other functions not related to their role as an oscillator that could affect how quickly the bacteria grows. So that's one problem. That's certainly one problem. Not having a clock is not really the same as not having at all Chi-C protein. That's a little bit different. What else? Let's say that as far as we can do, that's good. Okay, we can do knockout versus cells that have Chi-C. So let's assume that that's okay. What other problems, if any, might be in these experiments?
Is the growth rate a very good measure of selective function?
That's one thing, yeah. I mean, the growth rate, how quickly they grow, so they control for light and they try different things. That's not so bad, I guess. But of course, that's true that they provide nutrient all the time, and maybe if they do an environment where nutrient is limited, the response might be different. Okay, so that's one thing. Is the growth rate really a good measure? Maybe there is something else. So let's say growth rate is not so bad. It's one thing, maybe not the only one, but it's one thing that you can measure. If you want to study growth rate, what is still the problem, if any, of this experiment? If you want to study the selective advantage, when you say select, think of the meaning of the word selective. You want to study...
What is single strain?
Yeah.
They should separate them?
No. Yes, actually they separate them. This is no selective. That's the problem. I think that's the what you mentioned was very good. It's true. And what you said also was very true. One more thing that I noticed is that if you do single strain culture, there is no selection. They grow in an environment when there is only them. So they don't compete against anything. There is no natural selection so if they put both in the same medium when they compete for the same resource, then you might be able to make conclusions. So for example, if the strain with a clock, with the strain with Chi-C grows faster and outcompete the strain without, then you can say yes, obviously it's advantageous. So that's one problem. Okay, so now let's think about this one. And imagine we are all in a lab and we have unlimited funds and we have access to many things. And we want to do a similar experiment in eukaryotes. What experiment would you propose to test the selective advantage of having a clock versus not having a clock? I just want to get your ideas. Just completely, it's just like a brainstorming. It has been done. But I don't think you probably know. It was in Russia, actually. Well, it was in Russia, but by half of the team was in Russia and half of the team was in Groningen, which is a city in the north of Holland, in the Netherlands. So, anyway, you are studying eukaryotes, nothing with bacteria, and you want to make an experiment to analyze the selective advantage of the clock. Let's be more precise. You are having mammals. You are studying mammals. Say mice, for example. Actually, there is one reason why it was in Russia. I come to think about it. You'll never have it done in the UK. Never. It will be just broken by the Home Office. So that's true. No, you see why. You see why. So, Fayed, what experiment could you do? How could you test the selective advantage of a clock? Yeah, that's one thing you can check, the number of offsprings. Yes. But I want you all to be more precise. So you can check the number of offsprings, but under what conditions? What is the laboratory settings you have? And when you plan an experiment, especially with animals like a mouse, you must plan very carefully. What kind of environment? What do you provide? So think about. Yeah, this is one of the things you can check. You can check the number of offspring. Yes. So what kind of experiment settings do you see? Two strains. This is what 2 strains of mice. Yes. How do you mean you have to separate?
Separate, just like in this experiment, they don't let them to get in touch to respond.
Yes. So you cannot. Let's say you have 2 strains of mice, one with a clock, the other one without a clock, but you cannot separate them. If you separate them, it's also not good. So somehow they need to compete. But how do you make them compete?
With the reproduction, with checking the offspring, you could check if the offspring have the clock or not, and how many have the clock or don't have the clock. Because it means that they were more successful in passing on their genes.
But of course. So what do you mean? Do you breed a mouse with a clock with a mouse without a clock?
Let them breed as they like, and then see which. So you have them all together in one environment, and then you take the offspring.
What environment?
As natural as possible, but in a lab.
Okay. Remember, you have unlimited funds, and you can go to Russia. So yeah, so that's the thing. You need to, as I said, there are mice, laboratory mice, who have lost the ability to make melatonin. So obviously you don't really want to have inbred lines. It's not so good, right? You want to have something that is as wild as possible. So one thing to do is to provide a natural environment. So what people have done, and they went to Russia to do it, is actually they released mice in the wild. They released laboratory mice that had no clock in the wild. That's what they did. It was half wild, so they were in an enclosure where predators, like whatever birds of prey they were there, could access and kill the mice. So this is one experiment that they did. So you provide a natural environment, and you put 2 strains of mice that have or not a clock. So let's focus on have or not a clock. How do you do that? Have or not a clock with mice. How do you do that?
So how to make a mice without a clock?
Yeah, how to make a mouse without a clock.
Can it still survive without female? Can it still survive without clock and female?
They can still survive without clock and beamer. Well, yeah, that's one of the problems, yeah.
You could either, with the laboratory ones, remove that or affect the SCN.
Yeah, so you could make knockout animals. Knockout animals that, like BMAL1, have no more rhythms. But they also develop metabolic syndromes and all these things, and they get sick, their skin falls apart, they are very bad. So is it because of their lack of clock, or is it because they are roles of BMAL1? Same as we said before with Chi-C, maybe BMAL1 has more roles than just being a clock gene. So lacking a clock gene doesn't mean lacking a clock. One thing that you can do, as you said, is removing the SCN. And then the animals have no more rhythms, but obviously, yeah, so you have to operate different, even animals who have a clock, you have to operate them but not destroy the SCN. But you can do that. You can do. There will be problems, but they each provide different things. Okay. So you can operate 100 animals and destroy the SCN, and you operate 100 animals and you do the same thing, but you don't destroy the SCN. You just insert the needle, but you don't do anything with it. So this is one thing you can do. And if you have knockout mice, so the problem, as I said before, each protein may have many roles. So is it really only the lack of clock that contributes to difference that you observe, if you observe any difference? Or is it because they are the roles that you have affected? So what else could you do to sort of make your case?
I don't understand why we have to show the competitive advantage in this way. Why don't you just study those without a clock, and then you can find out various diseases, and then you can say that because without a clock.
Come on. Define your experiment. You cannot just say you just need to study.
No. I'm just saying that we are asking now. We're saying that we have to compare those without a clock and with a clock together, so that we can show that those with a clock have survival advantage.
No, you just want to show if there is any difference. You just want to show. You don't want to show the one with a clock have an advantage. You just want to know
there is a difference.
If there is a difference. Because if you want to show they have an advantage, then it's a very bad way to start an experiment. Because you are undermined by your own hypothesis. So you have to show, is there a selective advantage of having a clock or not? It's a very important question. Okay, so maybe there is no difference. But if the question you ask is, you can do, but it's a little bit dangerous, because then you will try to do an experiment to show that that they have a selective advantage. They may not at all have a selective advantage. But the point of the discussion is that it's very difficult, because one reason is that lacking a clock gene doesn't mean lacking a clock. Right? And destroying the SCN is also affecting potentially other things. But it has been done. It has been done. And there was no any clear things, probably because many things are. It's a compound thing, right? So it's a very difficult question to address. So that's why this paper about bacteria is a little bit more easy to do these things. Okay? So that's just the point of this discussion there. So let's go back to the knockout animals. And the one that like BMAL1, they are a little bit not so. So let's forget about what I said, that it has been done and everything. So the one lacking BMAL1, they are not so healthy. So what could you do?
Destroy the neurons that receive the light and transmit the signal into the SCN.
That's an interesting one. But what does it mean? What does it mean destroying the light input to the SCN? Does it mean lacking a clock?
It's not lacking a clock, but it means that the animals won't receive changes in the environment.
Yeah, that's an interesting thing. So the animals will have a clock, but will not be synchronized to the environment. So then, what do you think might happen? So the question you're asking is not having a clock versus not having a clock, is being in tune with your environment. And that's a very interesting question. It's not the question we were working on, but it's a very interesting question. So what is the selective advantage of being synchronized to your environment? So what do you think might happen? What would you predict? You are working with a nocturnal animal who somehow sees but doesn't have an input to the SCN. How would you do that? You don't want to study blind animals, because being blind doesn't mean not having the input to the clock. Being blind just means being blind. And a blind animal, you can expect, will probably die much quicker than an animal who isn't blind. But how could you specifically, I kind of want to integrate everything we have seen so far. How would you make an animal that isn't blind but has the input to the SCN destroyed or a bit weaker? How do you do that? Could you do that?
Does the rest of the brain communicate to the SCN? Or does it go that way?
Yeah, there are many connections. The SCN does a lot of connections everywhere.
So even if you can cut off the connection to the SCN, would the occipital lobe still give information to it?
So if you cut the connections,
So you cut the nerves that connected to the SCN, so the SCN itself wasn't interpreting the data from the eyes, but it still went to the optic nerve. The optic nerve then the optic lobe.
so let's so the way it goes, you have the eyes, then you have the optic nerves, you have the optic chiasm, and then the SCN sits on the optic chiasms, and then you have the brain, and the SCN goes to different parts of the brain, like for example to the pineal glands, and there is also a direct connection to the adrenal gland. So there are many connections. So what do you want to cut?
I think does the optic nerve also go to the? Isn't it processed in the?
Yeah. So the problem, if you cut the optic nerve, you will also cut every single visual image formation. So that's a problem. Okay. So you cannot just cut.
Is there any disease that causes the mice to not be able to perceive light, to perceive light but still not blind?
That's an interesting point. There are mice who are not visually blind, but they are not untrained to the light dark cycle. They can still see image, but they are not untraining to the clock. And this is because, and this is what I wanted to discuss today, it is because in the retina, there are different types of photoreceptors, which are the ones that are important for vision. Do you remember? Do you know already? In the retina, you have the rods and the cones that are responsible for image formation. But what about the clock?
The rods.
What about the clock? Not the rods. So the retina has many layers. Okay. The retina has many layers. Have you already learned about the photoreceptors that are important for the clock? And this is something I don't think we have discussed. Have you come across anything related to the clock? No? Hmm? Yeah, so that's your computer speaking, isn't it? That's not you, that's your computer. This you are cheating. This is cheating. This is cheating. Okay, so... Right, but you are right. So, well, your computer is right. So, which is different. So, in the retina, and I think I do have pictures for you, but sometimes this does not work. Try to get the slide for you. I will put these slides on classes, but I wanted to discover with you, otherwise you all know what we are going to speak about. Actually, maybe you already have these slides. I just need to check. Yes, actually it's there. And of course it's there we go. So what you have here is a cross section, well it's just a picture, but it's a cross section of the retina. And you have rods and cones which receive light and send to the optic lobe for visual information. Then you have bipolar cells, and in the inner region here you have the ganglion cells. And some of these ganglion cells have a different photoreceptor that is called melanopsin. And melanopsin is a specific kind of photoreceptor. Some of these retinal ganglion cells send information directly to the SCN for entrainment of the clock. And the way it works, you have the rod and the cones, and in the deeper layer, you have intrinsically light responsive retinal ganglion cells that express melanopsin. Not all of them. Some of them express the photopigment melanopsin, and they send information to SCN neurons. It's a glutaminergic innervation. And when these cells are stimulated, then you have all funny things happening with the signal transduction, which is calcium dependent. This activates kinase. This phosphorylate CREB. And do you know CREB? Have you come across CREB before? CREB is a transcription factor. When it is phosphorylated, it's activated, and it binds to CRE elements in the promoter of selected genes that have CR elements. And in the talk of the clock, the important gene is PER1. As you know, PER1 has an E-box, actually more than one, it has maybe 5 or something, and these E-boxes are important for the self-sustained oscillations of PER1 expression. But PER1 gene also has a CRE element in its promoter, which is responsive to CREB. So what does it mean? It just means that when you shine light at dark, you have a sudden burst of PER1 expression, which will then affect the clock. So this is how you reset the biological clock in DSCN. By acutely inducing the expression of PER1 gene via this mechanism, then you affect the phase of the clock. Okay. So to come back to our previous discussion, if you actually manage to remove melanopsin from these cells, then the input for the clock is much weaker. That being said, it's not completely disappearing. Even mice with no melanopsin in their retinal ganglion cells, they have still some indication that the light input to DSN is not completely disappearing. They are mostly free running, but in some cases you can still see some effect of the light-dark cycle because they have rod and cones and there are many connections between different. That being said, most of the rhythm is free running. Okay, so this is one of the things you can do. That was a good idea, to specifically remove the input to the clock. So the animals still have a rhythm. They don't have all these funny things happening when they don't have clock genes like BMAL1. They are not sick or whatever. They just have no input of the light axis. So that's one of the very interesting things you can do indeed. Okay, so that's just to say, this is from Travnikova Bendova paper in PNAS. So they show all the different inputs you can have to the per1 gene, how you activate per1 gene by light via. Okay. So that just explains what you say. So yeah, so maybe not destroying the clock, but destroying the light input. So what would you expect to happen? If you do that, you are not really studying the having a clock or not, you are studying, is it important to be in tune with the environment? So what data would you expect? For a nocturnal animal, suddenly the animal, what would you expect? In the light-dark cycle, what would you expect?
Animals with one type will be anxious. Wild-type animals will be anxious.
Wild-type animal will be advantageous. Yeah. You have to be careful. We all have to be careful, because what will happen is that there will be no difference whatsoever. Why? The animals lacking melanopsin, as I told you, they will be mostly free, their clock will be mostly free running. But I think you have forgotten something.
The environment is consistent. The environment is consistent.
The environment is so that you give a light-dark cycle to the animals, and the endogenous clock will be free running. So if you measure clock genes and everything, they will be free running, but so that means that if you put suddenly the animal in a constant darkness, you will see that the rhythm of the endogenous clock is completely out of phase with the environment. But there is still something that is important.
What period will change?
The clock period will be the same. The clock period will not change. It's only the entrainment of the endogenous clock will change. But there is something else which makes it even more difficult to study these kind of things. And it's something that maybe we saw very early in this lecture series. If you remember, one of the first things I showed you is that if you put the animal in a light-dark cycle, you have a rhythm, you have locomotor activity rhythm, you have a feeding rhythm. If you put the animal in a constant environment, you will still have a rhythm, but it will have a kind of slightly different angle, that corresponds to the length of the circadian period. And even if you have an animal that doesn't have a clock, like, for example, cry double knockout mice, which is, I remember, one of the slides that I put. Of course, in constant darkness, it has no rhythm. But do you remember the data in a light-dark cycle? What happened to a cry double knockout animal in a light-dark cycle? What did you get? What did you get? What was the data? If you put a BMAL1 knockout mouse or a cry1, cry2 double knockout mice, these are both arrhythmic in constant conditions, but what happened in light dark cycle?
Pretty much, they had a rhythm.
This is the same thing. They have a rhythm. Because this is a phenomenon called masking. light for nocturnal animal is acutely repressing activity, regardless of their clock. When nocturnal animals see light, they actually are afraid. They don't want to be active. So if you have an animal, even if they don't have melanopsin, they know it's light. So the locomotor activity is acutely inhibited by light. So that's a very difficult thing to do. It is a good idea, but because of this masking, we have no idea how this happens. We have no idea of the molecular players that actually underlie the masking of these nocturnal animals. But even though they have no melanopsin, under a light-dark cycle, they will be not active during the day, and they will be active at night, regardless of their endogenous clock. So that's another problem of studying these kind of things. Okay, and we have no idea how it happens. And that's the end of today's lecture. So it's very, very difficult thing to do. It's very difficult. This is why currently there is no really clear indication as to really is it an advantage or not of having a clock. Of course, we sort of say, yeah, if it's a nocturnal animal and then if it's there at the wrong moment, it's going to be eaten by, a predator. You can sort of imagine these things. You can sort of reason these things, but there are no currently very strong proof of that. It's just common sense, I suppose, but well, it's very difficult to design an experiment to really prove that that is the case because of many different things. Okay, so if you have any questions, please ask. Otherwise, it's the end of today's discussion. So I think you do have these slides, I think. In the first slide I put at the beginning of this lecture. So this, remember this? All these things that we saw before. And then we started to read papers after this point. But there are still some slides which I will go over also next week. All right? So there will be final exam in 2 weeks. Right? Okay.
What will we do next week?
I don't know yet. No, I will not see completely new things. Maybe a few new things, kind of interesting things which we have not seen yet, but not a complete. If you have questions, please ask. Review the material. If you have something you don't understand, ask questions. Make sure you understand everything that we have seen so far, and then that should be it. But we are going to see, like for example, or like things I want to see that I have not seen. For example, is chronochemotherapy, which is something I want to go over with you. We have not seen that yet. What could be the potential mechanisms of chemo? Have you seen this before? Have you heard this word? Chronotherapy. That's one of the. I think it's kind of interesting. So the fact that cancer treatment, if you actually take into account circadian time of the patient, the outcome of the treatment can be actually much better if you treat your patient at a certain time compared to other time. So we will see this. This is one of the things I want to see with you. Oh yes, I love questions.
Could you explain this figure?
Wait, so you weren't
Yeah.
there. You weren't there. So which A, B, C, or which figure?
This one.
This one.
Yeah. I don't know what this is.
Okay, so do you remember the previous paper we were looking, where they were doing chromatin immunoprecipitation?
Yes.
So they get the DNA that is bound by transcription factors, and if they have a lot of fragments from DNA, then it means the binding is strong.
Yes.
So this is the same thing. This is the same thing, but it's